This is a production of Cornell University.
All right. Hi, everyone. Thank you for staying to the very end. Hopefully I can entertain you with the final frontier, the Clavibacter-Solanum pathosystem. So in this talk, I'll give you a snippet of some of my dissertation research that has been unfolding over the past year. And I'm currently a fourth year PhD graduate student in Chris Smart's lab up in Geneva. So bacterial canker of tomato is caused by a gram-positive pathogen called Clavibacter michiganensis subspecies michiganensis. So this is a seed-borne bacterial pathogen that can spread in these greenhouse production facilities like I'm showing, through water dispersal, through mechanical inoculation, and can cause a lot of problems for growers across New York State and across the world. So once this bacterium is inside of the plant, it spreads through the vascular system, colonizing the xylem tissue, and in later stages of infection induces these stem cankers that you see up there, and as well as causing unilateral wilt and marginal leaf necrosis. During kind of later stages of infection as well, on the fruit it can form these bird's eye lesions that can reduce the quality of the fruit and also lead to economic loss. So this pathogen is especially important in New York because we're the #5 producer of tomatoes in the USA. And a lot of these tomatoes go to fresh markets, so they need to be blemish-free, can't have these lesions. And so some of the work we're trying to do is to support this industry in New York. Importantly, there's currently no known qualitative resistance to this pathogen. So I was interested in understanding tolerance to this bacterial pathogen, and we started by looking through the literature and finding that there were published tolerant accessions of Solanum hebrichides and Solanum arcanum, and I began screening them with both wild type and also a hypervirulent mutant of Clavibacter. And what you can see here is, I won't be able to see that, is the most susceptible Solanum lycopersicum cultivar mountain fresh, and also our most tolerant accession, Solanum arcanum 2157. And what we found is that the bacteria are able to colonize the tolerant accession to a pretty high density, albeit less than in the most susceptible plant. But it is impeded in its ability to spread through the vascular system vertically. Along with looking at kind of the vascular stage of this disease, we're screening these fruit to see if they're tolerant to the fruit portion of the infection. And so currently we have a lot of data for 2 of the 4 accessions, and including the susceptible Solanum lycopersicum and also Solanum arcanum 2172, different from the one I showed you before. And what we can see is that these bird's eye lesions form on all of the genotypes. And it's interesting because in this slide I showed you before, it was completely asymptomatic in the vascular stage. However, it's able to form lesions on the fruit in that asymptomatic species. So this has led us to some interesting hypotheses and directions where I'm taking this work that I won't be able to show you all the cool pictures I have. But we're looking at the host vascular morphology and how that impacts the ability for CMM to spread between xylem vessels and also vertically. So we're using a mix of confocal microscopy as well as scanning electron microscopy to see the colonization patterns of the bacteria and kind of focus in on individual xylem vessels and see how the bacteria are able to aggregate within those. We're also interested in looking at how the bacterium alters the host xylem environmental system, whatever. And we're using SAP metabolomics to try to understand that both in the tolerant and the susceptible species. So that work will be coming within the next year. So with that, I'd like to acknowledge my lab and everyone that has participated in this project and helped out a lot. And Mary Jean Welser helped me with the SEM, and I didn't put up there, but I'd also like to thank the Rick Tomato Genetics Research Center and also USDA Grant in Geneva for providing tomato germplasm. So I think we have a couple seconds for questions, and thank you. Yes. Yes, so the hypervirulent strain of Clavibacter. So that's a knockout of an expansin-like gene from the bacterium. So it's a secreted expansin protein or expansin-like protein that the molecular mechanism that is still unknown. All right, thank you.
This has been a production of Cornell University, on the web at cornell.edu.